So I wanted to start off by thanking Lucas and Noldus company for inviting me to chair this, and also all of you all for coming, and my two other speakers, Yetta and Garrett. I'll start by introducing myself, telling you a little bit of background about myself and the topic I'm going to talk about today, motion and emotion in the basal ganglia. Can everyone hear me in the back? Microphone's working? So I've been studying the basal ganglia, and specifically a structure in the basal ganglia, the striatum, for about 10 years. In graduate school, I was studying this in the context of drug addiction, giving rats cocaine, looking at the firing of nucleus accumbens cells. And then I went on to a postdoc in San Francisco at the Gladstone Institutes, and I was then again studying the striatum, but in the context of Parkinson's disease. And something that always was a little unsettling to me was when I was studying drug addiction, I learned that the striatum is involved in processing emotions in the reward associated with drugs of abuse and withdrawal states associated with withdrawal from drugs of abuse. And then in my postdoc, I learned the basal ganglia is actually involved in movement, in organizing and executing motor patterns. So I was trying to bring these two together and thinking, what does it do? Does the striatum do reward, like the addiction literature tells you, or does it do movement, like the Parkinson's literature would say? And at some point, I started to think that these things don't separate completely. I'm not sure they separate at all. And really, you never see emotional states without a motor component. What does it look like when someone's angry? What does it look like when someone's sad? And by the same token, you don't often find, or many motor states are also associated with an emotional state. So what does it feel like for you right now sitting in a chair? And would it feel different if you were running? Would it feel different if you were picking up a heavy box? These movements have feelings associated with them. I'll show you one example of this link, with apologies to our Dutch hosts. I'm going to show you the last 10 seconds or so of the World Cup Championship game in 2010.
No.
So note the time, we're at extra minutes, 1:16. So the Spanish team scores, and they immediately race to the side of the corner of the field, jumping up and down, pleased with their performance. And now again with apologies, we're going to watch it one more time. We're going to watch it a little slower, and I want you to focus on the Dutch players. We're going to watch their motor response. So here's the goal, and they freeze. So So if we knew nothing about the game of soccer, and we were watching just the motor behavior, which is in some ways the situation that I think a lot of us find ourselves when we're looking at our mice, you can come up with a conclusion based on the motor behavior that following the goal, the Spanish players are more active than the Dutch players. And if you like quantifying behavior by video, you could even quantify this. Now the meters moved in the 3 seconds following the goal by the average Spanish player on the field and the average Dutch player on the field. I showed this video at a talk in France, and a very astute audience member asked me, well, how much did the referee move? So it turns out that he moves about 3 meters, not a clear preference. We're almost done with the game. So because this is a game of soccer, and we've all seen it or played it, and we understand the emotions that are associated with this game, we know that there's more than motor effects in these players following the goal. These are 2 photos taken about 1 second and about 2 seconds following the goal. And we know the emotional states that the Spanish players and the Dutch players are in. And this is really the outline of what I'm going to talk about today, is that there's 2 populations of striatal neurons that exert an opposing control over motor output. And I'm going to show you this to you in a fairly convincing way. And then I'm going to try to convince you that these same populations have an opposing control over emotional states, meaning the cells that make us move also make us feel good, or associated with those states. Cells that make us slow down and stop movement are also associated with negative emotional states. I use the word convinced because it's a little harder to get into conclusively discussing emotional states in mice. Let's start with a brief overview of striatal anatomy. So here we're looking at a coronal section of a mouse brain. I don't know if you can see this down there, but this histology is courtesy of collaborator of mine, Chip Gerfin at the NIH. Just wanted to point that out because I put this a little bit low down here. So we're looking at a coronal section of a mouse brain, and I'll tell you about the fluorescence in 1 second, but just to orient you, this big area on top is the cortex, and then the striatum is rather large in the mouse, about 20% of the volume of the brain. It's this huge structure sitting underneath the cortex. So the fluorescence here, this mouse has been injected with a virus that targets 2 different fluorophores, red and green, to the 2 main neural types in the striatum. So if we zoom in on here, you'll see that there's 2 main populations of neurons. They comprise about 95% of the striatum. If you watch the red and green go off, they're distinct. These are known as the direct and indirect pathway neurons because of their projection pathways, which I'll show you in this video. So we're now going to be looking backwards through the brain following these projections. So this is the coronal section at the level of the striatum. We'll move towards the back of the brain, and you'll see where these red and green cells project. The green cells are sending fibers out here. They synapse in a structure known as the pallidus. And if we keep going back, red fibers come right by the pallidus and come clear across the brain and end up in a structure known as the nigra. If we put all this sliced histology data together into a 3D reconstruction, you can see this really nicely. So up here in the striatum is where the virus was injected, and then there's fibers that come from the indirect pathway neurons and synapse in the pallidus, and fibers that come from the direct pathway neurons and synapse all the way across the brain in the nigra. I'm going to call these 2 populations direct pathway medium spiny neurons, or they'll be abbreviated with a D, DMSNs, or indirect pathway abbreviated with the I, IMSNs. I'm going to introduce one final technical thing, which is optogenetics. And the data I'm going to be showing to you is expressing optogenetic proteins in these pathways using techniques that are very similar to how these fluorophores were expressed. The channel that you'll hear about from me and also my co-speakers is channel rhodopsin 2. This is a light-activated cation channel that has similar currents to an AMPA receptor, but instead of being activated by glutamate, it's activated by blue light. So seeing that here, that this is a patch recording of a cell, and every time a blue light is flashed, the cell fires an action potential. So we wanted to do this in our direct and indirect pathway neurons, both to look at the effects on movement, and then later I'll show you the effects on the emotional state. This is some histology of viral-mediated expression of channel rhodopsin in these 2 populations. Here we're looking at a sagittal section, similar to the final position of that 3D movie, where the striatum's up here, the pallidus is next to it, and the nigra across the brain. If we now express just one fluorophore, with channel rhodopsin tagged with YFP, you can see these fibers, similar to the movie in the direct pathway, come all the way down to the nigra. In the indirect pathway, they stop right next door. To show a little bit more specificity, now we're looking at the nigra itself. And this has been co-stained for TH, which is the tyrosine hydroxylase marks dopamine neurons. And we see that these, in the direct pathway, these green fibers expressing channel rhodopsin are coming into an area known as the reticulata that's mainly GABAergic cells. And then in the indirect pathway, none of these green fibers make it here. They're terminating in the pallidus. So now we can do experiments and look at the effect of stimulating these pathways on motor output. And I'm just going to jump to the punchline. I'm going to show you a video showing you optogenetic stimulation of indirect pathway neurons, and that it has a strong inhibitory control over movement. In this video, there's a few features I'm going to call your attention to. One is the word laser will turn on when the laser is actually on, stimulating this pathway. And then the mouse has 2 tracking features. There's a red dot tracking the center of the mouse, and there's going to be purple showing pixels that are changing from frame to frame. I'm going to use these 2 tracking features for some of the quantification in the next few slides. Oh, so bad. What do you guys think? Should I find this video?
Yes.
All right, let's do that.
Yeah, please.
mouse running around, when the laser turns on to stimulate these, I think watching the purple really gives you an idea of how much it shuts down his behavior, how much it can shut down depending on how strongly you stimulate. We're going to see one more pulse that'll be a little bit longer. So this is going to be about 15 seconds. For reasons that I forget, I've done these for 5-6 minutes, and it's pretty much the same, that he'll stay frozen. Once it finally turns off, he shakes it off in a couple seconds, and he's back to normal. So we can quantify this in a few ways. I mentioned we're going to use the center point or the total activity. Using the center point, we can quantify our, I'm going to split this behavior into 3 things. Using the center point, I can quantify walking. Basically, if the center point is moving from place to place, means the animal is actually going somewhere. We can also use the purple pixels changing to quantify freezing. As you saw, if they're freezing, the mouse is frozen, it goes down towards 0. And then everything else, I'm kind of lumping in as fine movement. The mouse does a lot of things that are not walking and they're not freezing. He's grooming, he's sniffing. And at the moment, they're pretty difficult to tease apart with automated methods, so I'm just calling them fine movement. What we're looking at here is the percent of time, so it adds up to 100%. He's only doing these 3 the way that I'm splitting the behavior up. We're looking at 30 seconds before the laser, 30 seconds during the laser pulse, or 30 seconds following. What you see when we stimulate the indirect pathway is that freezing goes way up. As you saw in that video, they spend more than half their time frozen while the laser's on. They don't walk very much while the laser's on. And then fine movement, the amount of time they spend grooming and sniffing, is also reduced simply because they're frozen. If we do the same thing with the direct pathway neurons, we get the opposite effect, that they almost don't freeze while the laser's on. They spend a lot more time walking. And then again, the fine movement, the sniffing and grooming, is reduced simply because they're now moving around the bowl and walking around more than fine movements. We can get a little deeper on this behavior by analyzing some finer aspects. So here what I'm showing you are 3 things. One of them, this is looking at walking now, is the frequency that they walk. The next one's going to be the duration they walk, so every time they make a movement, how long is that movement? And finally, the speed that they're walking. So every time while they're walking, are they moving with a different motor pattern, essentially. What we see here is that this is all normalized to 1, being the speed when the laser's off. With the indirect pathway stimulation, they initiate less frequently, so the movements are a little bit shorter, and they're slower. If we do this for the direct pathway, see that they initiate a lot more frequently. This is sort of the main effect, that a normal mouse walks around like kind of walking and freezing and walking and freezing. These mice, as soon as they stop, they go again. So they end up with a lot more movements. They're slightly longer, and interestingly, they're pretty much the same. If we look at the freezing and fine movements with a similar analysis, now we're looking at the frequency of freezing. With the indirect pathway, they freeze a little bit more frequently. But the main thing, as you saw in the video, is once they stop, they're stopped. So the length of each time they stop is way longer. And then fine movements, which is really the vigor that they're doing the grooming and sniffing and everything that's getting sorted as fine movement, is reduced similar to how the ambulation movements are reduced when the indirect pathway is stimulated. For the direct pathway, we have the opposite, that they, as I mentioned a second ago, they freeze much less frequently, and once they freeze, they move right away again. And in this case, the vigor that they're doing their grooming and sniffing is actually increased. They do this with a higher speed. I'm going to show you one more piece of data. This is something new from my lab at NIH. This is data collected by Kim LeBlanc and Danielle Friend, where we're testing whether these movement effects are light dose dependent. Dose dependent doesn't really make sense with light, but basically doing a dose response curve and seeing if can we stimulate more. And there's something surprising that came out of this. So the way that we ran this experiment was we picked 8 doses of light, 0 to 2 milliwatts. And we ran them in trials that were 30 seconds on and 60 seconds off. So I'm going to show you the aggregate of 3 trials each at each power. And for each mouse, we randomized this, such that this is sort of a schematic. of, imagine these mice going on and on. But each mouse got a random presentation of these powers. And this allows us to do something nice that's not to look at time with the light off versus the light on, but actually look at time at 0 milliwatts, sort of a fake light pulse, and compare that into it. It's sort of a fairer comparison to compare these doses head to head. And the surprising thing that, for me, that came out of this was the indirect pathway. We're now looking at light power down here, and I'm just showing you speed of the mouse. I'm not getting into all those finer movement parameters. But it saturates pretty low at 0.25 milliwatts. Remember before, I may not have mentioned this, before I was doing everything at 1 milliwatt, you can really get very low light powers. Some of these powers down here, this is 50 microwatts. It almost can't see it. So you look at the mouse and you're like, is it on? It's really small amounts of light can get these pretty robust behavioral effects. We did the same thing with the direct pathway. It's a little more linear, that we don't really start to see anything until about a half milliwatt, and it seems to keep going up. So it may be that this reflects some difference between these pathways, that the indirect pathway, just how it's wired up, may be a lot more sensitive and can shut down the behavior at levels of firing that are lower than that in the direct pathway. One question you might be having is what kind of effect these low light powers have on the firing. We've done in vivo recordings with this. We put electrodes in and we shine the light at various light powers. This is just a summary figure of 4 different light powers. So we have 100 microwatts, 300 microwatts, 1 milliwatt, and 3 milliwatts. So we actually get pretty robust increases in firing even at these very low power stimulation. So hopefully I've convinced you of this first part, that these direct and indirect pathway neurons have an opposing control over motor output. The indirect pathway shuts down the mouse. This motor movement, the direct pathway, facilitates it. And now I'm going to try to convince you that these same populations exert an opposing control over emotional states. And right away we come to something that many people in the room grapple with, which is, how do we know the emotional state of a mouse? I think there's sort of several logical ways through this question. My personal opinion is that we really don't know. Nobody knows what it feels like to be a mouse except a mouse. That's just my opinion. Some people feel differently. However, we can take logical approaches through this and say, for instance, one way was to say, we can't know, but we can get clues about the mouse's emotional state from its behavior. So we can do things like reinforcement and punishment. Reinforcement has come from the psychology literature. Reinforcement describes increases in behavior. Punishment describes decreases in behavior. And we know that for ourselves, things that we find reinforcing are often linked to positive emotional states. Things that are punishing are often linked to negative emotional states. If you try a new food and you like it, you're likely to eat more of it. If you try food and you then eat less of it, you probably didn't like it, that kind of logic. But really this doesn't rely on understanding the mouse's emotional states. And this was the first approach that we took for getting into this question. I do think you can also study emotional states more directly, but I'm not going to show you data on that in this talk. So the way that we did this for reinforcement was you put the mouse in a box and hooked it up such that he had 2 triggers he could touch. One of them was linked to a 1-second laser pulse, the other one was linked to no consequence, just to kind of figure out how much the mouse likes interacting with these triggers. Essentially, this was taking me out of the loop. In the first part of the talk, I was controlling the laser. Now let's let the mouse control it and ask, do you want to stimulate the direct pathway or the indirect pathway? I'm going to show you a video of the direct pathway stimulation. It turns out they like this a lot. These are actually the 2 triggers down here. You can see them. The one on the left is hooked up to a 1-second laser pulse. The one on the right, nothing happens. And this video was taken about 10 minutes into the mouse's training. So 10 minutes ago it was naive. These mice picked this up very quickly. I'll mention the video is sped up 10 times. It's not a So to quantify this and how quickly they learn, now we're looking at expressing this as the percent of laser peri-trigger contacts from 0 to 100, although they don't get up to 100. Meaning that if you were to only press on the laser trigger, that would be 100%. If you never press on that, that would be 0%. And we see over the first 30 minutes of training, these mice learn this very rapidly. For people that train mice, this was amazingly fast for a mouse to learn anything. If we bring them back for 2 more days of this, they remember it at the beginning of the day, and they seem, at least with this limited amount of training, to be asymptoting around 75% of their touches. This is only 90 minutes of total training, so I don't know possibly it could go much higher. When we did the same thing with the indirect pathway, we got the opposite effects, where they learn very rapidly to avoid touching this trigger that results in a 1-second pulsed laser to stimulate the indirect pathway, that pathway that froze them. They didn't like that being stimulated. And then they sort of remember it, but not as well as the direct pathway, something I'll show in the next experiment. If we quantify this kind of to get at this effect, we're looking at the first 2 minutes. This is now presented in bar graphs, just the first 2 minutes of each day. We see that on day 1, they're at 50%. They haven't figured it out in the first 2 minutes. By day 2 and 3, the direct pathway, they remember this. The indirect pathway, it's a little bit lower in magnitude, and by day 3, this is not even a significant aversion. I'm going to show this sort of difference a little bit more clearly in another experiment. Now we're doing a a real-time place preference or avoidance. In this experiment, the mouse, if he goes on one side, he will get laser stimulation. If he goes on the other side, he's safe. And we present the data pretty similarly, the time spent on the illuminated side. And we see that across 2 days, the mice learn to spend, they're getting their direct pathway stimulated, they learn to spend more time over there, they remember it, and spend more time on day 2. Indirect pathway, they learn to stay away from it. They kind of have to relearn it again on day 2. And then this experiment, we did an extinction test, where we now just turned the laser off and said, where do you want to hang out? Do you want to hang out on the previously illuminated side or not? And for the direct pathway, they did. They wanted to hang out where they used to get the light stimulation, even though they're no longer getting it. For the indirect pathway, it's like they forget that they used to not enjoy being there immediately. It's really right away when we turn it off, they're back at 50%. So I think this does reflect some difference in the plasticity mechanisms that govern each pathway. That postsynaptically depolarizing the direct pathway can have this long-lasting effect on behavior and presumably on those cells or their inputs, whereas the indirect pathway, this postsynaptic depolarization is not sufficient for a change in behavior. I would guess that it's not sufficient for a change in the plasticity onto those cells as well. So I'm going to conclude here. The 2 summary points of what I've hopefully showed you are that these striatal direct and indirect pathway neurons have an opposing control over motor output. And then these same pathways have an opposing control over emotional states, representing a neural link between movement and emotions. I'd like to acknowledge everyone who contributed to this work, especially the Kravitz lab. This is my postdoc lab where most of these experiments I showed were done, as well as my lab at NIDDK, Danielle, Kim, and Kadia. And I'll leave these conclusions up. I'd be happy to take a few questions. And if you think of any at a later date, my email's down here, lexkravitz@nih.com. Thank you.
Lex, do you have any idea about the cell types that drive the behavior in direct or indirect pathway? Can you take it about that?
You want to repeat the question?
Which cell types?
Are normally driving?
Yeah.
So the question was on which cell, I'm going to ask first. The question was which cell types are driving these behaviors normally. Maybe which cell types are synapsing onto them to drive the direct or indirect pathway neurons. So I don't know if that, I'll repeat, I'll ask the question that I'm going to answer, and then she can ask a different one if that's not it. So Yeko is asking, hopefully, about, under normal conditions, who's driving these pathways, what's making them fire and promote or inhibit movement. And they receive input mainly from cortex and thalamus as excitatory input that'll drive them, but they have a very important modulator that also acts on these cells, and that's dopamine. The one distinction between these direct and indirect pathway neurons is they have a different dopamine receptor. The direct pathway has a D1 receptor, which is excitatory. The indirect pathway has a D2 receptor, which is inhibitory. So when you think about dopamine's effects in the brain, dopamine comes from down here, comes back to the striatum, and it'll excite the D1 pathway, facilitating movement, facilitating the positivity. That's that way. At the same time, dopamine can be inhibiting the indirect pathway, so essentially turning off the brakes on movement and also turning off the brakes on these negative emotional states.
Can you speculate why the indirect pathway is actually the relatively short? Basically,
The question was on why the indirect pathway actions were so quick, why the effects were so immediate.
no, actually, you're showing that effect is quickly forgotten.
Oh, yeah. So the question was essentially why the indirect pathways effect is quickly forgotten, and I can speculate on that. So when you watch these mice doing this task, one of the strangest things is they don't like it. They go over there, they get the stimulation, they run away, and then 30, 45 seconds later, they're like, hey, I don't remember that. Let me see what's in this room. And they do that for the entire 30 minutes. So normally, if this was a shock or something, they would do that once or twice in the beginning, and then they would just stay away. They would have a memory of it. It's almost like the thing happens, the memory is it's 30 seconds long, and then it's like it didn't happen after that. So that's what it looks like when you watch them. So I think about the plasticity mechanisms onto these cells, the main thing being cortical fibers coming in, and the synapses they're going to form onto these striatal neurons. And what I would think is happening is that for the direct pathway, those synapses are getting strengthened. There's something has changed. In the indirect pathway, it goes away. It may be a very short-term plasticity that is not maintained after 30 seconds. It's like the event didn't happen.
So in a lot of the labs that try to look at emotional states, anxiety, depression, we often have to control for locomotion. So if you're going to do an open field, an elevated plus maze, you have to show that they have no differences in locomotion. If we take that motor response is innately related to emotional state, would you say that is still a valid control? And if it's not, would you have any ideas on maybe what might be a better control?
So the question was that often when you're studying depression or negative emotional states, you have this compound of movement. And I guess your question is whether that's a valid, or how I'd sort of address this compound.
Well, is it a valid control if I have? But should that be something that you have to make sure is the same in your groups, or one group is if a negative emotional state will innately be lower moving, should that not have to be shown?
So my opinion is that a negative emotional state will have a lower amount of movement. So that's seen in people. There's a whole host of very interesting literature on depression and the motor symptoms of depression that include postural changes, gait changes, changes in the speed, like things that change the speed of saccades and arm movements. So I really think if you're going to study a disorder and then try to find a time when that symptoms of that disorder are not happening, you may be shooting yourself in the foot. This is a symptom as much as the negative emotional state itself.
Hi, nice talk. Have you considered measuring 70 kilohertz ultrasonic localizations?
Yes.
What have you found?
Oh, so I've considered that. The question was whether.
So you considered it but not done it, okay.
Yes. I think that's a very interesting idea, and yeah, we're going to do that pretty soon. All right, so I'm going to move on. Again, please feel free to email me or find me later if you have any more questions.